Hey, before we start the video, if you like this sort of content and you want more, come check out Med School Bootcamp. We have content for Step 1, Step 2, Anatomy, and OMM. Right now, we're about to watch the Biochemistry section. And if you're looking for downloadable PDFs, along with shorter videos and quizzes after each video to help you reinforce the information, then you can find it all on Med School Bootcamp. We also have a Step 1 question bank with some of the most representative questions to help you prepare for Step 1. You can also create a custom Step 1 study schedule when it's time for dedicated. So if you're looking to crush it in med school this year and pass Step 1, come check out Med School Bootcamp. It's going to be your first link down in the description. Now on to the video.
Hi everyone, my name is Paige and thank you so much for joining me for a chapter on oxidative stress. This is a short chapter, but a fun one. We're going to start by talking about the pentose phosphate pathway and producing NADPH. Then we're going to move into one of my favorite topics, which is the oxidative burst. And this is going to be kind of a biochemistry, immunology crossover episode. And then we will finish by talking about ethanol metabolism. Let's get started with our lesson on the pentose phosphate pathway. This is also known as the HMP shunt or the hexose monophosphate shunt. And the point of this pathway is going to be to generate NADPH. We've talked about NADPH a couple of times in previous chapters, but basically this is really important in anabolic reactions as well as in oxidative stress. It is a supply of reducing equivalents in steroid synthesis and fatty acid synthesis. It's also used in reducing glutathione and the oxidative burst and overall management of reactive oxygen species. The other thing that this pathway produces is ribose 5-phosphate, which is implicated in nucleotide synthesis. And this pathway is all happening free of ATP usage. So no ATP is going to be used. It's also not going to be produced. And this is all going to happen in the cytosol. So let's take a look at our image here. Over here in yellowy orange, you're going to see the glycolysis pathway that should be beaten into your brain by now. And then in purple coming off the side here is going to be the HMP shunt, right? Or the pentose phosphate pathway. So we're going to start this process the same way we started glycolysis by turning glucose into glucose 6-phosphate via the enzyme hexokinase. And we formed this glucose 6-phosphate intermediate of glycolysis. And we've established that yes, this can go on to become fructose 6-phosphate and continue all the way on down glycolysis and turn into pyruvate. But this intermediate can also take a little bit of a side quest out here, and that side quest is the pentose phosphate pathway. And the first reaction of this side pathway over here is going to be favored when there is a lot of G6P available. The implication there is that there's a lot of glucose if there's a lot of glucose 6-phosphate, which means that we sort of have extra glucose to play around with. We're not sacrificing anything from glycolysis necessarily in producing this pathway. So when there's high G6P, we're going to do the Step 1 right here, turning glucose 6-phosphate into 6-phosphogluconolactone. And the enzyme that mediates this is really high yield. It is called G6P dehydrogenase. And to make this conversion, this G6P dehydrogenase is going to take NADP plus and turn it into NADPH. Now, one thing I really want to make very clear is that NADPH is not the same as NADH. They are both sources of reducing power. They are both electron carriers, but they are used in totally separate processes. So just be very mindful about that. You can remember the P in pentose phosphate pathway or HMP shunt makes NADPH. Then we're going to take that 6-phosphogluconolactone, combine it with some water, and turn it into 6-phosphogluconate via the enzyme 6-phosphogluconolactanase. And then once we've made that 6-phosphogluconate, we can use enzyme 3 over here, which is called 6-phosphogluconate dehydrogenase, to form ribulose 5-phosphate. And this is another step that's going to load electrons onto NADP plus and form NADPH. So now we've had two different NADPH producing steps. And additionally, this step is going to spit out some CO2 as well. Now, all of the enzymes that were specific to the pentose phosphate pathway up until this point, so 1, 2, and 3, have all been irreversible. So this glucose 6-phosphate is effectively committed to the pentose phosphate pathway through these irreversible reactions. So let's go ahead and take inventory of these irreversible reactions back on our outline. The first step is something you're familiar with, you've seen 1,000 times, glucose turning into glucose 6-phosphate via the enzyme hexokinase. That's the first step of glycolysis. Boring, we've seen it 1,000,000 times. However, when that glucose 6-phosphate is high, we are going to favor this side pathway here, this pentose pentose phosphate pathway. And we are going to utilize the enzyme G6PD to commit this G6P into the pentose phosphate pathway and turn that G6P into 6-phosphogluconolactone. And this is the rate determining step of the pentose phosphate pathway. And we made our first source of new reducing power, NADPH, at this step by turning NADP plus into the NADPH. And remember, we can remember that the source of reducing power we're making is NADPH because it is the PPP pentose phosphate pathway. The next step was pretty uneventful. We turned that 6-phosphogluconolactone into the 6-phosphogluconate via the enzyme 6-phosphogluconolactanase, and that required 1 molecule of water. And then we got back to the good stuff with another interesting step where we made another NADPH and we released some CO2. We did that by turning 6-phosphogluconate into ribulose 5-phosphate via that enzyme 6-phosphogluconate dehydrogenase. Now let's talk about our reversible reactions, which is the second half of the pentose phosphate pathway. We can use enzyme 4 right here, which is called phosphopentose isomerase, to turn ribulose 5-phosphate into ribose 5-phosphate. One thing you'll remember is ribose 5-phosphate is the starting substrate for nucleotide synthesis. It can turn into PRPP, which can then go on down the line, eventually split into IMP and UMP to make pyrimidines and purines. And this is all outside the scope of this video, but it is just important to be aware of where that starting substrate comes from because you have seen it before. And if we're not using that ribose 5-phosphate for nucleotide synthesis, we can use enzyme 5 here called transketolase that has a vitamin B1 cofactor. We can use that to facilitate the reversible reaction that breaks ribose 5-phosphate into fructose 6-phosphate and glyceraldehyde 3-phosphate, and both of those are glycolysis intermediates. So if we have a bunch of extra ribose 5-phosphate, we don't need to turn all of it into PRPP for purine and pyrimidine synthesis. We can actually just send it back over into glycolysis. It just starts as glucose, goes out, takes the scenic route. What's needed is sort of picked off, and then the rest can go back in and rejoin glycolysis. And remember, this is reversible, so technically we can take these two glycolytic intermediates and combine them together to form ribose 5-phosphate. So back to the outline, our first reversible reaction was turning ribulose 5-phosphate into ribose 5-phosphate via phosphopentose isomerase. Then we turn that ribose 5-phosphate into those two glycolytic intermediates, G3P and fructose 6-phosphate, via that enzyme transketolase, and that had a vitamin B1 cofactor. And we said that ribose 5-phosphate was also a starting substrate for nucleotide synthesis, everybody's favorite topic. So we already talked about why we need the ribose 5-phosphate, but we only kind of loosely touched on why we need the NADPH, right? What are we making all this reducing power for? If your test is tomorrow and you can only remember one thing about NADPH, remember that it is used to help with oxidative stress. What I mean by that is it is specifically used for glutathione reduction within red blood cells, and reduced glutathione can detoxify free radicals. So free radicals are a major cause of oxidative stress, go around wreaking havoc in your body. Reduced glutathione can be used to calm down those free radicals. And we reduce that glutathione with the electrons loaded onto NADPH. So we want to make reduced glutathione. Reduced glutathione is also called GSH. And oxidized glutathione, or not reduced glutathione, sort of our starting point, is also called GSSG. So through a simple oxidation reduction reaction, we can take those electrons off of the NADPH, load them onto the GSSG, and form the reduced glutathione, GSH right here, leaving behind NADP plus in the process. And the enzyme that does that is called glutathione reductase. So that's a high yield point. That's something that you want to lock down. The reason that we want to make this reduced glutathione, this GSH, is because when combined with hydrogen peroxide, we'll turn that hydrogen peroxide into just plain old water, which is not toxic and not a free radical. And we reform that oxidized glutathione in the process. So we're back to where we started. We're going to need another NADPH to reduce that glutathione so that it can be used for another detoxification reaction. And the enzyme that's running that reaction is called glutathione peroxidase. And these are both important reactions, and they're cool reactions we're going to see when we talk about the oxidative burst next. And I got a little ahead of myself with that, but the creation of reactive oxygen species inside phagocytes can kill bacteria. So phagocytes in your immune system can actually create their own reactive oxygen species, right? Harness that destructive power to destroy dangerous bacteria. But you can't just leave the destructive free radicals floating around when you're done, right? We need to kill the bad guy without killing ourselves. And so we need that NADPH to subsequently neutralize those free radicals when we're done with them to spare that phagocyte. That's coming up later this chapter. Another use of NADPH is in cholesterol synthesis. So the rate-limiting enzyme of cholesterol synthesis is called HMG-CoA reductase. This turns HMG-CoA into mevalonate, and we'll talk about that more when we talk about cholesterol. But for now, just understand that the rate-limiting step of cholesterol synthesis requires NADPH. NADPH is also used in fatty acid synthesis. And the step we're looking at is the step mediated by the enzyme fatty acid synthase. And that fatty acid synthase enzyme is going to add 2 carbons onto malonyl-CoA to elongate that fatty acid chain. And that step, mediated by fatty acid synthase, also requires NADPH as a reducing agent. So you're noticing a theme here of anabolic reactions. And then finally, the cytochrome P450 system also needs the electrons from NADPH for cytochrome P450 reductase to do its activity. So lots of cool stuff we need NADPH for. Finally, let's talk about a clinical implication, and that is going to be a G6PD deficiency. So remember, G6PD was that rate-limiting step. It was that irreversible step that committed that glucose 6-phosphate into the pentose phosphate pathway. When we are deficient in G6PD, enzyme 1 here, we're not going to run that step, and we are not going to form NADPH. So no G6PD means no NADPH. And remember here, no NADPH means that we cannot form reduced glutathione. And no reduced glutathione means that we can't handle free radicals. We can't break them down and detoxify them. What that means is, in times of stress, when there are increased reactive oxygen species, patients with a G6PD deficiency cannot protect against those reactive oxygen species. They're vulnerable, and they will develop a hemolytic anemia where their red blood cells just start to explode from the stress. And you can check out the HEMOC section if you want more details on that, because that is a very important deficiency to be aware of. So let's wrap things up. We've been talking about the pentose phosphate pathway, also called the HMP shunt. They mean the same thing. We created 2 big products with this. The first was ribose 5-phosphate, which we used for nucleotide synthesis, and the second was NADPH, which was used in a bunch of anabolic reactions as well as oxidative stress. We started the way that we start glycolysis by turning glucose into glucose 6-phosphate, and then when that G6P was high, we had lots of G6P left over, we could send it on a little side quest down here to form some of these important extra products. The big enzyme that we talked about was the first enzyme, which was irreversible, and the rate-limiting step, that was G6PD. And during those irreversible steps, we were able to form NADPH, that reducing power. With that ribulose 5-phosphate that we made, we could enter the second half of this pathway, those reversible reactions. We could turn the ribulose 5-phosphate into ribose 5-phosphate for nucleotide synthesis, and then whatever was left over, we could just send right back into the glycolysis pathway. We talked about why NADPH in particular is so important for a series of anabolic reactions like cholesterol synthesis and fatty acid synthesis. We also talked about its implication in the cytochrome P450 system, but by and large, the big takeaway for NADPH is that it helps detoxify free radicals by reducing glutathione. So if we can't make NADPH, for example, in the case of a G6PD deficiency, patients with that are going to be much more vulnerable to oxidative stress and may suffer from hemolytic anemia, for example. And we started thinking about how we can use that theme of neutralization so that we can potentially harness the power of reactive oxygen species, because we can then control it and neutralize it down the line. And that's what we're going to talk about next when we talk about the oxidative burst. Until then, that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige, and thank you so much for joining me for our lesson on the oxidative burst. It's also known as the respiratory burst, and it's actually an immunology topic, but there's just enough biochemistry sprinkled in there that they let me do it. I think this is one of the coolest topics. So we're going to be looking at phagocytes, and specifically what's happening inside of a phagolysosome. When a phagocyte has captured a bacteria, it has to find a way to kill it. And I just finished telling you all about the dangers of reactive oxygen species in the last video on the pentose phosphate pathway. But your phagocytes are going to just spit in the face of danger. They're actually going to create reactive oxygen species, and in doing so, they're going to create a biological weapon to aim at and kill and destroy these bacteria. So the first part of this burst is going to be to create reactive oxygen species within a phagocyte to help the immune response. The second part of that then is, now that we've unleashed the beast, those reactive oxygen species, we are actually going to have to neutralize them to make sure that they only kill the bacteria and don't kill your cell in the process. And so that NADPH that we just finished creating in the pentose phosphate pathway is going to be critical for both halves of this, right? Both the production of the reactive oxygen species and then the neutralization when we're done. So this in blue out here, right, is our phagocyte, and it is phagocytosing a bacteria. And so this sort of bleb in here is the phagolysosome that is basically gobbling up and eating the bacteria. And so we're going to start off looking inside the phagolysosome as to how that killing actually occurs and that generation of the reactive oxygen species. Big shocker to you, I'm sure, is that in order to generate reactive oxygen species, we have to use oxygen. We're going to use enzyme 1 right here, which is called NADPH oxidase, and we are going to use the NADPH that we made in the pentose phosphate pathway to turn that oxygen into a free radical called superoxide, which is oxygen with an unpaired electron. Then we're going to take enzyme 2 called superoxide dismutase. We're going to take that superoxide and turn it into peroxide. You can see now we've sort of reached a fork in the road here with what we can do with peroxide. We're going to continue on down the path for now, and we'll come back to those side paths in a minute. But right now we're focused on our end goal of just killing the bacteria. So we're going to take enzyme 3 here called Myeloperoxidase, and we are going to turn that peroxide, combine it with a chloride to turn it into hypochlorite, which is HOCl, again with an unpaired electron. Now, interestingly, some invading bacteria can actually produce their own hydrogen peroxide. And when those bacteria release that inside the phagolysosome, they're just going to enter the pathway right here and continue on down the line. So your phagolysosome is equipped to handle that peroxide being made by these combative bacteria. And finally, we're at the part where we get to kill the bacteria. We're going to take that hypochlorite with its dangerous unpaired free electron, and we are going to kill the bacteria, split it right open. Now let's go back to peroxide and talk about these two other branches here. I just finished telling you that invading bacteria produce their own hydrogen peroxide, and that your phagolysosome can harness this in addition to this whole oxidative burst pathway in order to kill the bacteria. However, some bacteria have an enzyme called catalase. This is a property of the bacteria, right? This is not intrinsic to your phagolysosome. The bacteria have this with them, and it makes them more resilient because they're able to neutralize their own hydrogen peroxide into water and oxygen. And so this means that your phagolysosome cannot turn your bacteria's hydrogen peroxide that they made back against them. They're able to neutralize that. And that'll be important in some pathology we'll talk about at the end of the video. All right, so we did it. We killed our bacteria. Fantastic. Now we have these reactive oxygen species floating around in the phagolysosome, and like I said, we can't let it stay there or it's going to destroy the phagocyte. So that hydrogen peroxide is going to come into the phagocyte, and that phagocyte is going to use an enzyme we talked about when we talked about the pentose phosphate pathway last video, this enzyme 4 here, which is glutathione peroxidase. So remember, reduced glutathione was a really special product that could detoxify peroxide. When you take that reduced glutathione, combine it with the peroxide, you can do a redox reaction. In the process, you will form oxidized glutathione, and that peroxide will turn into water. So when I was first learning this content, I did not understand the difference between catalase and peroxidase. Both of them are taking peroxide and turning them into something harmless. Broadly, the difference between a catalase and a peroxidase is that catalase is going to use 2 peroxides, and it's going to just sort of shuffle the oxygens around and form water and O2. A peroxidase is going to take 1 peroxide and then another source of reducing power and make different products. So instead of 2 peroxides, it's 1 peroxide and 1 something else. In this case, the something else is reduced glutathione. And we're still forming water, but then our other product is going to be the reformation of the oxidized glutathione. So we just talked about peroxidase, which formed water. But we had to sacrifice a reduced glutathione, which became oxidized glutathione. And so we want to find a way to reform that reduced glutathione so we can go on and detoxify more peroxide. Remember, we reform reduced glutathione by using the reducing power from NADPH. The enzyme that does that is enzyme 5 right here, glutathione reductase. And all that step 6 here is showing you is just that G6PD step, right? That first rate-limiting step of the pentose phosphate pathway. That was the first NADPH producing step of that pentose phosphate pathway. So that's where the NADPH came from. So our neutralization reaction is happening at this step here with enzyme 4, glutathione peroxidase. Steps 5 and 6 here are just showing how you regenerate that reduced glutathione, right, via NADPH, and where that NADPH is coming from. It's reminding you how all of this ties in to the pentose phosphate pathway, which is in this purple box right here. All right, take a breath. Let's recap on the outline here. Remember, the first step of the oxidative burst was turning O2 oxygen into superoxide, which was that oxygen with an unpaired electron. The enzyme that did that was NADPH oxidase, and that required NADPH from the pentose phosphate pathway. We took that superoxide and turned it into peroxide via the enzyme superoxide dismutase. We combined that peroxide with a chloride, and we formed hypochlorite, and the enzyme that did that was myeloperoxidase. Now, this myeloperoxidase enzyme contains heme. It also has a blue-green pigment, and that is why when you cough up sputum when you're sick, it's the myeloperoxidase enzyme that gives the sputum its, blue-green color. And then some side points we made about peroxide: invading bacteria could generate peroxide by themselves, and it would just sort of enter the pathway here at this step, and the phagolysosome could take that peroxide and turn it against the bacteria. But those extra sturdy bacteria that have that enzyme catalase, we call those catalase positive organisms, they had that bacterial catalase enzyme that could neutralize the peroxide into water and oxygen. So your body isn't able to turn their own peroxide against them. And then in that last step, the hypochlorite, which is basically bleach is what hypochlorite is going to go and kill that bacteria, and it's going to promote the release of lysosomal enzymes. And now the phagocyte has to find a way to neutralize all that extra peroxide. It's going to do that by using the enzyme glutathione peroxidase to combine that peroxide with reduced glutathione to turn that peroxide into water and then oxidize the glutathione. And so at this point, we've successfully carried out our neutralization reaction, but we need to remake that reduced glutathione for the next neutralization reaction. That's what these next two steps were about. We reformed that reduced glutathione by that enzyme glutathione reductase, and we reduced it with the reducing power from NADPH from the pentose phosphate pathway. And one of those steps in the pentose phosphate pathway that we were interested in was that G6PD rate limiting step. And like we talked about in the last video, if you're deficient in G6PD, you're not going to be as robust against oxidative stress because you're not able to neutralize those reactive oxygen species as efficiently because you can't reduce the glutathione to participate in that detox reaction. So, all super cool. Let's talk about clinical implications. A condition that you are very likely to be asked about when you're being asked about this whole process is one called chronic granulomatous disease, or CGD. And this is when your patient is born without NADPH oxidase. So step number 1 right here is knocked out in chronic granulomatous disease. What that means is these patients' phagolysosomes cannot do the oxidative burst. They can't turn oxygen into superoxide, but they still have another trick up their sleeve. They can still use bacterial peroxide. Remember, I told you, invading bacteria can generate their own peroxide, and your phagolysosomes can turn this peroxide against them without necessarily having to do all the beginning steps of the oxidative burst. Okay, cool. So hope is not lost. Except if these patients get infected by catalase-positive organisms. Remember, those catalase-positive organisms could detoxify their own peroxide. What that means is they're going to go ahead and break down their own peroxide that they came in releasing into water and oxygen, and the phagolysosome isn't going to be able to turn it against them and use it and kill the bacteria. The bacteria is going to get ahead and neutralize it itself first. So these patients can't do the oxidative burst, and then when a catalase-positive organism is there, and remember this is specific to catalase-positive organisms that the second part applies, that peroxide that's being generated by the bacteria can't be used by the phagolysosome because it's going to get neutralized before the phagolysosome even has a fighting chance. That means that patients with chronic granulomatous disease are at an increased risk for infection by catalase positive organisms. And this is why, and this is a very high yield point. They want you to not only understand this fact, but understand why it works. And there's all of this potential for higher order questions of, then can you remember which organisms are catalase positive? So there's lots of sneaky ways they can try and ask about this pathway, both from a biochem standpoint, from an immunology standpoint, and from a microbiome standpoint. So be on the lookout. So these patients are at increased risk for pyogenic infections with these catalase positive organisms. And as a refresher, some of those are Staph aureus, E. coli, Pseudomonas, Serratia, Klebsiella, Nocardia, Candida, and Aspergillus. And one little courtesy thing they might give you in the STEM to try and tip you off to the fact that this is in fact chronic granulomatous disease is they might talk about a negative nitroblue tetrazolium test. And so basically that's just looking for the activity of NADPH oxidase. When NADPH oxidase is there, normally this colorless dye that they put in to do the test for will turn black. If there's no NADPH oxidase, the test is going to be negative because there's going to be no enzyme activity for a color change. And finally, let's talk about an especially dangerous bacteria, one of my favorites, Pseudomonas. Specifically, we're going to be talking about one of the virulence factors of Pseudomonas aeruginosa called pyocyanin. And I'm going to do this in a blue-green color because this has a blue-green pigment. And this pyocyanin virulence factor is going to give Pseudomonas a really cool competitive edge against some other bacteria in the area. And it's going to generate reactive oxygen species. So what it's going to do is it is going to utilize NADPH to start reducing oxygen and turning it into superoxide and hydrogen peroxide, reactive oxygen species. That's going to force the phagocyte to work really hard and have to waste a lot of its reduced glutathione just trying to keep up with all these reactive oxygen species that the Pseudomonas is throwing at it. However, not only are we chewing up all of this reduced glutathione just because we're producing a ton of reactive oxygen species, the pyocyanin is also going to directly oxidize the reduced glutathione into oxidized glutathione. And that oxidized glutathione can't do anything. We need it to be the reduced form in order for it to detoxify reactive oxygen species. So we're getting this buildup of reactive oxygen species and this real depletion of the reduced glutathione that you need to detoxify the reactive oxygen species. And that is a winning recipe for Pseudomonas, thanks to that pyocyanin virulence factor. So let's wrap this all up. We've been talking about what happens in phagocytes, specifically within the phagolysosome that those phagocytes create in order to chew up bacteria. Those phagocytes are going to laugh in the face of danger, and they are going to create reactive oxygen species in order to kill those bacteria. Of course, when they're done, they then need to make sure that they neutralize those reactive oxygen species that they've been harnessing for killing power to make sure that they don't accidentally go ahead and kill themselves. And both the bacterial killing pathway and the neutralization pathway required NADPH from the pentose phosphate pathway. We started with oxygen and went through a series of steps where we eventually formed more and more powerful free radicals till we eventually ended up with bleach that could just bleach and kill the bacteria. One of those intermediate steps in there was peroxide, and we also talked about how invading bacteria can make their own peroxide, but the phagolysosome is smart and can turn that peroxide against the bacteria, unless it's a catalase positive organism. And catalase positive organisms are going to beat your phagolysosome to the punch and neutralize their own reactive oxygen species before it can be turned against them. Now that we've made all those reactive ROSs, it's time to shut them down and neutralize them. And we did that with the enzyme glutathione peroxidase, which turned that peroxide into water with the help of the very important substrate, reduced glutathione. And NADPH was also really critical for the formation of that reduced glutathione, and that again comes from the pentose phosphate pathway. Then we talked about some cool clinical implications, the first one being in chronic granulomatous disease. These patients could not perform the first step of the oxidative burst mediated by NADPH oxidase, which was fine until they encountered a catalase positive organism. And those catalase positive organisms were self-neutralizing their own peroxide, leaving these patients with CGD effectively defenseless against catalase positive organisms. And then we talked about pyocyanin, which was a virulence factor of Pseudomonas aeruginosa, and how it created all sorts of different reactive oxygen species to outcompete all of its bacterial competitors. And that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on ethanol metabolism. So when you drink an alcoholic beverage, the alcohol that is in that beverage is ethanol. And of course, it is no surprise to anyone that that ethanol is not very good for you. So your body needs a way to metabolize that ethanol, and that's what we're going to be talking about in this video. And we're going to be talking about two pathways of ethanol metabolism today. The first one is sort of the standard pathway that happens in the cytosol, and this is just happening when there's sort of a normal amount of ethanol for the body to handle. However, in heavy ethanol consumers, there is a backup pathway that can get recruited, and that is a pathway that happens in the endoplasmic reticulum, and so we'll talk about that pathway as well. So like I said, that cytosolic pathway is the major pathway of ethanol metabolism. It's going to handle about 90% of the ethanol metabolism in low or moderate ethanol consumers. So let's get oriented to our diagram. We are looking at a liver cell, a hepatocyte. So this is our hepatocyte. We've got the endoplasmic reticulum over here. We've got the cytosol all out here in purple. This is the mitochondria, and then here is the cell membrane, and then out here we've got a blood vessel. And our first step here is to take that ethanol and break it down into acetaldehyde. And acetaldehyde is this intermediate. And so there are two pathways, like I mentioned, to break that ethanol into acetaldehyde. There's a cytosolic pathway, and then a pathway that passes through the endoplasmic reticulum. And then once that acetaldehyde is formed, both of those pathways kind of merge, and then the final step is the same. So let's talk about the cytosolic pathway, and the enzyme that's going to turn the ethanol into the acetaldehyde here is called alcohol dehydrogenase, and this is a very important high-yield enzyme. The important thing to understand about alcohol dehydrogenase, or ALD, is that it is going to take NAD+ and it is going to reduce it to NADH by loading it up with electrons. And this is interesting because a lot of pathways that we've talked about so far sort of use NADH, use the reducing power that NADH holds. This is the opposite. Here we're actually producing that NADH, and that's going to have some consequences down the line. And so that's the major pathway and the major enzyme, alcohol dehydrogenase, in patients who are maybe ethanol naive. However, in heavy alcohol consumers or patients with alcohol use disorder, this endoplasmic reticulum pathway is going to kick in and play a larger role. Here, ethanol is still turning into acetaldehyde, but it is going to require the cytochrome P450 system, specifically CYP2E1. And that enzyme's living on the surface of the endoplasmic reticulum. And in the process, we're taking NADPH, not NADH, so different from the first step, NADPH, and we're turning it into NADP+. And the formation of this NADP+ is going to promote the creation of reactive oxygen species. Remember, NADPH is protective against reactive oxygen species. And this reaction is really chewing it up and turning it into NADP+. And additionally, other sort of factors of this reaction are also promoting the formation of reactive oxygen species like superoxide. The pathway mediated by alcohol dehydrogenase in the cytosol is the major pathway. So we're not going to get a whole lot of these dangerous reactive oxygen species. However, once the level of consumption becomes heavier and we have to recruit this overflow pathway here, that's where we're really going to start to see that buildup of reactive oxygen species, and that is destructive. So now, through one pathway or another, we formed our acetaldehyde. That's an intermediate, and that is a toxic intermediate in and of itself. We don't want to just leave this acetaldehyde floating around. So we're going to break that acetaldehyde down into acetate, which is a ketone, and the enzyme that we're going to use to do this is aldehyde dehydrogenase. And just like alcohol dehydrogenase, aldehyde dehydrogenase is going to turn NAD+ into NADH. So now we've formed two molecules of NADH, and you can see how this NADH might start to build up pretty quickly. That acetate, like I said, is now a volatile ketone. It can go out into the blood and then even out to the breath. So let's go back to our outline here and fill in a couple of those gaps now that we have our framework. So we'll start by talking about the cytosolic pathway. That cytosolic pathway was mediated by alcohol dehydrogenase and turned ethanol into acetaldehyde. And this reaction, remember, required NAD+ to turn into NADH. One thing you might hear talked about a lot with alcohol dehydrogenase is enzyme kinetics. So alcohol dehydrogenase participates in zero-order kinetics. What that means is it is going to be eliminating a constant amount of alcohol per unit time. Remember, zero order is a constant amount per unit time First order is a constant fraction per unit time. So we're doing zero order, a constant amount per unit time. And so you've heard the rule of thumb that your body can metabolize 1 drink an hour, regardless of how much is in your system. That's where that's coming from. And the NADH that was made during this step, remember, was made in the cytosol. It's going to need to go into the mitochondria where it can actually be used. And so that's going to participate in the malate aspartate shuttle, which we have a whole other video on in a previous chapter. And remember that acetaldehyde intermediate is toxic. It's going to form DNA adducts, and that is carcinogenic. Additionally, it can cause vasodilation, which can cause things like nausea and facial flushing. And that's something I want you to remember for now, because that's going to come up a couple more times later in our discussion, what happens physically to you when you get that buildup of acetaldehyde. Then we shifted our attention over to that overflow pathway that was happening in the endoplasmic reticulum microsomes. That was where the cytochrome P450 isoform CYP2E1 was mediating that same reaction of ethanol to acetaldehyde with a couple of twists. Remember, normally this is a minor pathway. It's only about 10% of ethanol metabolism in low ethanol consumers, but this pathway is ramped way up in heavy ethanol consumers. The NADPH here is getting its electrons from the cytochrome P450 pathway, specifically the enzyme NADPH cytochrome P450 reductase, and that's in the ER membrane. That was what provided the electrons for the NADPH to participate in this reaction. And the reason why this pathway is ramped up in heavy ethanol consumers is because it has a high Km, right, which means a low affinity for ethanol versus alcohol dehydrogenase. The kinetics don't favor this pathway unless the other pathway is already full because of that low affinity. One thing to really be aware of here is that when this pathway is ramped up, we're ramping up that CYP2E1 enzyme, and this is not the only metabolism reaction that CYP2E1 participates in. Famously, CYP2E1 promotes the metabolism of acetaminophen into NAPK, which is a toxic metabolite. And this buildup of the NAPK is going to suck up a lot of reduced glutathione, right? Reduced glutathione being our warrior against reactive oxygen species. So it's going to make those patients more vulnerable to reactive oxygen species. And that is why patients with alcohol use disorder or who are very heavy alcohol consumers should try and stay away from acetaminophen because it can become very hepatotoxic very quickly. We're ramping up that CYP2E1 enzyme, then you go and take acetaminophen, and it's going to metabolize that acetaminophen more into another hepatotoxic substance. And like we talked about, this pathway also promotes the formation of reactive oxygen species like superoxide and predisposes patients to oxidative stress. Then we talked about that common pathway of, okay, what happens after we formed this toxic acetaldehyde intermediate? It's going to turn into acetate via the enzyme aldehyde dehydrogenase. It's another reaction that requires NAD+ to turn into NADH, and then that acetate can go out of the cell and into the blood. Something I want to talk about here quickly is what would happen if aldehyde dehydrogenase was defective. Well, if we can't break down acetaldehyde, we're going to get a buildup of acetaldehyde. So what are some examples of when acetaldehyde dehydrogenase would be defective? Well, one example is that there is a mutation where you have decreased ALDH activity. What that means is you get a buildup of acetaldehyde, and remember that acetaldehyde causes vasodilation. So patients with this mutation may have a lower tolerance to alcohol because they'll start to experience facial flushing or nausea. Additionally, some cells, like cells in the esophagus, naturally have a lower ALDH activity. And remember, acetaldehyde is carcinogenic. And so that is why patients who are chronic consumers of ethanol may be predisposed to esophageal cancer. Finally, there are drugs that can actually inhibit aldehyde dehydrogenase. For example, the drug Disulfiram. Basically, it is for patients with alcohol use disorder in order to try and create an aversion to alcohol. It's going to block out this step. You're going to get a buildup of the acetaldehyde, and then those patients are going to get that nausea and the facial flushing, those unpleasant vasodilatory features, because they can't break that acetaldehyde down into acetate. And so that's actually one of the clinical implications of this pathway, is that Disulfiram drug, where it is going to inhibit ALDH, which will promote the buildup of acetaldehyde, and that will promote those unpleasant features that eventually lead to alcohol aversion. Now, this drug itself isn't used so much because compliance is pretty difficult for patients. No one wants to take a drug that makes them not feel good. You're more likely to hear about Disulfiram in the context of Disulfiram-like reactions. And these are reactions that can happen with certain drugs, like, for example, metronidazole, which is an antibiotic that causes this disulfiram disulfiram-like reaction. So when patients drink alcohol on metronidazole, they're going to experience a very similar phenomenon to what they would experience if they were taking disulfiram. They're going to experience that nausea and that facial flushing the same as if they were taking disulfiram. And that's what a disulfiram-like reaction is. Another huge, big, important, high yield, star, bold, underline, clinical implication of this is a fact that we've already touched on, which is that an increase in ethanol consumption is going to lead to an increase in the ratio of cytoplasmic NADH to NAD+. And there are a ton of implications for this. The good news is, if you've been following through this biochemistry unit chronologically, a lot of these are already going to make sense to you. So brace yourselves, I'm bringing back some old diagrams and we're going to walk through exactly what I'm talking about. This is going to be a review, but it's important to understand all of these in the context of ethanol, because really they could test you on any one of these pathways and you're supposed to understand the implication of the increased NADH that the ethanol is providing to them. Let's start all the way on the right here. This is from the video where we talked about the fates of pyruvate, and we're going to look at this reaction right here mediated by enzyme D-lactate dehydrogenase. This was a bidirectional enzyme where we could take pyruvate and combine it with NADH to turn it into lactate and NAD+. So when NADH is high, we're favoring that forward reaction where we turn pyruvate into lactate. This is going to lead to a lactic acidosis. Next, we'll come over to the left here and we're going to take a look at the TCA cycle. And you'll remember the steps in the TCA cycle, like isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase, were inhibited by NADH. That means that high NADH is going to shut down the TCA cycle. So if we shut down these steps, we're eventually going to get a buildup of acetyl-CoA. And now let's shift over to our metabolism map that you guys are familiar with by now to look at the implications of that buildup of acetyl-CoA. We'll zoom in down here around the TCA cycle, and we already talked in a very early video about alcoholic ketoacidosis. So when the TCA cycle is shut down and that acetyl-CoA is building up, and that acetyl-CoA building up is also compounded by the fact that acetate, which remember was the end product of that ethanol metabolism, can also turn into acetyl-CoA. If we can't go down around the TCA cycle, that acetyl-CoA is going to have to take a detour, and it's going to take a detour down into ketone metabolism and is going to form ketones. So not only did we form that acetate, which is a ketone, but because we shut down the TCA cycle and caused a buildup of that acetyl-CoA, that led to alcoholic ketoacidosis. And additionally, that's going to put this patient in a low energy state. We're going to get a buildup of AMP because we can't create ATP by the TCA cycle. All right, now things start to get a little tricky. We're going to slow it down a little bit. We're going to look at the malate aspartate shuttle next. And specifically, we're focused on sort of the form of the malate aspartate shuttle that was happening in the fed state. The reason that we're focused on this is because the fed state is sort of induced when levels of NADH are high. It's like a mimic of glycolysis having happened, because glycolysis also creates NADH. And so don't even look at this bottom stuff for now. We don't even really care about that right now. We're focused on the top part and the conversion between malate and oxaloacetate. All of that extra NADH that gets formed is going to participate in the malate aspartate shuttle, where those electrons are going to get loaded onto malate, brought into the mitochondria, and then released inside the mitochondria. So what that means is that when NADH in the cytoplasm, which is out here, this is the cytoplasm, right? This was the mitochondrial matrix. When NADH in the cytoplasm is high, we're promoting the conversion of oxaloacetate into malate. And the enzyme that did that was malate dehydrogenase. And so keep that in your mind, is when NADH is high, we're promoting the conversion of oxaloacetate to malate in the cytoplasm. Now let's take a look at gluconeogenesis. One of the steps of gluconeogenesis in the cytoplasm, right here now we have our mitochondria, out here is the cytoplasm, and one of those steps was turning malate into oxaloacetate in the cytoplasm. So we just finished talking about how in the fed state, in the cytoplasm, we want to turn oxaloacetate into malate. And for gluconeogenesis, we need the opposite to occur. So we are favoring the reaction that is the reverse of gluconeogenesis. We're getting a decrease in gluconeogenesis because we have too much NADH in the cytosol. Another reason why we are getting a decrease in gluconeogenesis is because remember I told you the TCA cycle was shut down and we got that buildup of AMP, right? It's important to note that this is sort of a pseudo-fed state. It's not a properly fed state. We just have this artificially inflated NADH. And remember, gluconeogenesis requires an actual high energy state. It requires ATP to run. We need to invest energy into that process. So because we have high cytosolic NADH and we have high AMP, both of those factors are going to decrease gluconeogenesis. And that is why you get hypoglycemia with prolonged alcohol use. Okay, that was the hardest one to wrap your head around. We're gonna come back to the metabolism map. The last clinical implication we'll talk about is alcoholic fatty liver disease, right? Hepatosteatosis. We're gonna zoom in again right into the middle of our map here. We've already set the stage here that the TCA cycle is shut down, so we got that buildup of acetyl-CoA and we were looking to divert it into other pathways, right? That was how we ended up with the alcoholic ketoacidosis. But you'll notice another pathway that it can shunt into is the fatty acid synthesis pathway. We can take that acetyl-CoA, turn it into malonyl-CoA, and turn it eventually into fatty acids and triglycerides. And that's not a pathway we've covered yet in the biochemistry unit, so you're just going to have to take my word for it. You're also going to see increased triglycerides for a second reason that we haven't talked about, which is increased activity of the glycerol 3-phosphate shuttle. And that's occurring at this intersection right here on the map, where we have this glycolytic intermediate, DHAP, that can turn into glycerol 3-phosphate by that G3P shuttle. And turning DHAP into G3P requires NADH. So when NADH is high, it's going to favor the formation of that glycerol 3-phosphate. And as you can see, that glycerol 3-phosphate can also turn into triglycerides. So that's two mechanisms for increased triglycerides in the liver. And that is hepatosteatosis, right? Fatty liver. Okay, that was draining. Let's go back to the outline and just take a summary of what we just talked about. First, we talked about the fates of pyruvate and how the high NADH was going to promote the formation of lactate and decrease the conversion of lactate back into pyruvate, and that led to lactic acidosis. We talked about how that decrease in the TCA cycle, because some of those enzymes were inhibited by NADH, as well as the increased acetate conversion to acetyl-CoA, favored a buildup of acetyl-CoA that needed to shunt over into other pathways, and that led to alcoholic ketoacidosis, as well as a buildup of AMP, right? And that did some weird things to gluconeogenesis. So we had the high AMP plus the high NADH led to a decrease in gluconeogenesis because of our low energy state, but our malate aspartate shuttle was tricked into thinking we were sort of in the fed state, and those all clashed and, shut down gluconeogenesis, and that was why we saw hypoglycemia. And then just like that buildup of acetyl-CoA caused ketoacidosis, it could also shunt into fatty acid synthesis to form triglycerides, and that was compounded by the increased activity of the G3P shuttle, which was fueled by that high NADH, and that led to hepatosteatosis. The last thing we will talk about is much simpler, and it's a topic you're already familiar with from last chapter, and that is the metabolism of methanol. Here I just want to bring your attention to the fact that alcohol dehydrogenase is not only used in ethanol metabolism, it is also used in methanol metabolism, as well as the metabolism of ethylene glycol. But alcohol dehydrogenase has a much higher affinity for ethanol than it does for methanol. What that means is that if a patient consumes methanol, which is going to be broken down into these toxic metabolites, they're at risk for methanol poisoning. If we give the patient ethanol, right, just good old-fashioned couple shots of whiskey, it will distract this alcohol dehydrogenase enzyme because that ethanol has a much higher affinity. It's going to snatch that alcohol dehydrogenase activity away from this methanol metabolism. What that means is that the methanol won't be broken down into those active metabolites. And so while yes, there is a drug called fomepizole, which is an alcohol dehydrogenase inhibitor, you can use alcohol in an emergency first aid setting to basically have the same effect. With the fomepizole, we're directly inhibiting it. With the ethanol, ethanol, we are effectively distracting the enzyme. And alcohol dehydrogenase is the first enzyme in ethylene glycol metabolism as well, so it's the same principle for ethylene glycol poisoning. The increased affinity of alcohol dehydrogenase for the ethanol is going to allow ADH to be inhibited and have a very similar effect to fomepizole. So we are ending this chapter with a bang. Let's sum everything up. Ethanol, as much as you might love it, it is toxic. We need to metabolize it. There are two pathways to metabolize it. The first one happens in the cytosol. That's sort of the major pathway that you're going to encounter in people who are ethanol naive or moderate ethanol consumers. That uses an enzyme called alcohol dehydrogenase, which will create NADH in the process. And that step happens via zero-order elimination kinetics. This step forms acetaldehyde, which is carcinogenic and also has vasodilatory effects, which can be pretty negative. However, there was also an overflow pathway that happened in the endoplasmic reticulum. The enzyme that mediates this pathway is the cytochrome P450 isoform CYP2E1. It also participates in the degradation of acetaminophen into a toxic metabolite NADPH, which is hepatotoxic. And this CYP2E1 activity also increases reactive oxygen species and puts the body under oxidative stress. So once we had used either of those two pathways to form acetaldehyde, we turned that acetaldehyde into acetate via aldehyde dehydrogenase. This was another reaction that produced NADH. Then that acetate could go out into the blood. NADH shuts down a whole bunch of processes. It helps to explain why you see some of the clinical features of prolonged alcohol consumption. Lactic acidosis due to decreased conversion of lactate to pyruvate, alcoholic ketoacidosis due to shutting down of the TCA cycle, hypoglycemia due to the shutting down of gluconeogenesis, and hepatosteatosis due to the buildup of triglycerides from the increased fatty acid synthesis. We talked about disulfiram and the disulfiram-like drug reactions, which happened when aldehyde dehydrogenase activity was decreased. You got that buildup of acetaldehyde, which led to those vasodilatory effects like the facial flushing and the nausea. And then we talked about how whiskey is good for first aid. If your patient happens to have methanol or ethylene glycol poisoning, we can use that alcohol to distract alcohol dehydrogenase away from those metabolic reactions and save your patient from poisoning. Okay, you did it. That's all I got for you for this chapter. If you have any questions, comments, concerns, problems, troubles, worries, hopes, dreams, fears, send them on my way. Otherwise, good job, good luck, and keep practicing. All right, go ahead, take a minute to read through the question, and then we'll come back together to talk about the answer. All right, so we have a 37-year-old man with no significant past medical history, and he's brought into the emergency department by a friend due to confusion and lethargy. And the friend is reporting that the patient has been refusing to eat for the past 2 days, and he's been complaining about some nausea and vomiting. And the patient is not able to provide a clear history. Now, when they take the patient out of the history-taking equation like that, it's usually intentional, so you're going to have to fill in the gaps using the physical exam and the labs. He's afebrile, his blood pressure is a little soft, he is tachycardic and tachypnic, and he's disheveled with dry mucous membranes, but no evidence of trauma. So we know at this point he's dehydrated and we don't have a ton of history from him. But when they start painting this picture of this disheveled guy and they kind of take a lot of the history-taking element away from you, you start to really think about substance use. The question writers are forcing you to maintain your index of suspicion for that based on this picture that they're sort of painting for you. When I start to go through the labs, I see that we have an incomplete ABG, which is kind of nice because there's not a whole lot we're expected to do with it. But we have enough information to tell that we have a pretty significant acidosis, right, a low pH, and that acidosis has at least a metabolic component. We can tell that because the bicarb is low. And we saw that he was tachypnic, so he's probably trying to compensate for that acidosis by trying to blow off some more CO2, which is acidic. We're going to see a slightly elevated lactate, a low glucose, and a very elevated serum beta-hydroxybutyrate. That beta-hydroxybutyrate is telling you that you have ketoacidosis. But be careful because not all ketoacidosis is diabetic ketoacidosis. We'll work through our differential for ketoacidosis and what that serum beta-hydroxybutyrate could mean in a minute, but let's finish the question first. We're also given that salicylates are negative and a urine drug screen is negative. And the question wants to know which of the following mechanisms best explains the metabolic derangements seen in this patient. In other words, what is the process that is underlying this patient's metabolic acidosis with components of both ketoacidosis and mild lactic acidosis and hypoglycemia? So when we see an elevated serum beta-hydroxybutyrate or ketoacidosis, we should have a framework. My checklist is diabetic, alcoholic, starvation, pregnancy, and miscellaneous, which the big one for this question, since we sort of said we were worried about substances and overdose, would be salicylates. And so to make a bit of a quick aside, we don't know necessarily that this patient has a high anion gap metabolic acidosis, although we can pretty much assume. But I know that everybody loves the mud piles mnemonic for the causes of high anion gap metabolic acidosis, and the D in mud pile stands for DKA. But I would challenge you to take a step back and make sure that you're including all of the causes of ketoacidosis in your differential there. All right, now we know that our male patient is not pregnant, and we're given that salicylates are negative already, so that's off our differential. And that really leaves us with diabetic ketoacidosis, alcoholic ketoacidosis, and starvation ketoacidosis. And when we see ketoacidosis in combination with a low glucose hypoglycemia, that should be screaming alcoholic ketoacidosis, right? In DKA, diabetic ketoacidosis, you're going to see high serum glucose. And in starvation ketoacidosis, as long as all of your other metabolic pathways are intact, you shouldn't see any change to the serum glucose at all. Your body should be cranking up other pathways to keep your blood sugar stable. All right, so we think our patient has alcoholic ketoacidosis. And those findings are characterized by an increased ratio of cytoplasmic NADH to NAD+ in hepatocytes. In our lesson about ethanol metabolism, we talked about how a lot of these reactions in breaking down ethanol use NAD+ to make NADH. And in biochemistry, we have a lot of reactions whose directionality is influenced by the ratio of NAD+ and NADH. So changing that ratio is going to have a lot of metabolic consequences. Increased NADH is going to increase the conversion of pyruvate into lactate, and that forms a lactic acidosis. Additionally, several TCA cycle enzymes are inhibited by an increase in NADH. So with increased NADH, we're going to shut down the TCA cycle. Additionally, in ethanol metabolism, we break down our ethanol into eventually acetate, and that acetate turns into acetyl-CoA. So we have a ton of extra acetyl-CoA, but it can't go into the TCA cycle. So instead, it is going to shunt over into other metabolic pathways, like ketone production, causing ketoacidosis. Additionally, our NADH to NAD+ ratio is going to disfavor gluconeogenesis. And so this leads to hypoglycemia. So therefore, the clinical profile here matches alcoholic ketoacidosis, which is answer choice C. So let's go through the incorrect answer choices. Answer choice A, an absolute deficiency of insulin, would be representative of diabetic ketoacidosis. In DKA, we see hyperglycemia, not hypoglycemia. The deficiency of insulin means we can't bring serum glucose into the cells. So instead, it just floats out around in the serum. So the opposite of answer choice A would be hyperinsulinemia, or too much insulin. And that's actually represented by answer choice E here. Suppression of gluconeogenesis, indeed, we do see in alcoholic ketoacidosis. But lipolysis is increased in alcoholic ketoacidosis because of counter-regulatory activation like catecholamines and glucagon. In hyperinsulinemia, we see suppression of both gluconeogenesis and lipolysis. And so while we would see hypoglycemia in hyperinsulinemia, we would not see ketoacidosis. Insulin typically prevents ketoacidosis by inhibiting lipolysis and ketogenesis. That's why you see ketoacidosis in a deficiency of insulin, in diabetic ketoacidosis. All right, answer choice B, glycogen depletion in prolonged fasting, is representative of starvation ketoacidosis. Like we said, in starvation ketoacidosis, we shouldn't see hypoglycemia. Other processes like gluconeogenesis and ketone metabolism should be keeping our blood sugars in check. Hypoglycemia is unique to alcoholic ketoacidosis among these differentials. Answer choice D, increased tricarboxylic acid activity, is incorrect because we already talked about how the TCA cycle is decreased, it's inhibited in alcoholic ketoacidosis. Some of those TCA cycle enzymes require NAD plus to be activated and will be inhibited by the excess NADH. So D is not correct. Answer choice F, toxic uncoupling of oxidative phosphorylation, in the case of this question is representing salicylate toxicity. We don't have enough information in the ABG to see if there's a mixed acid-base disorder going on that would point us towards a salicylate toxicity, but at any rate, we would not see hypoglycemia in salicylate poisoning, plus we were told the salicylates were negative. So this is out too. So in summary, we talked about our framework for ketoacidosis, which is indicated by an elevated serum beta-hydroxybutyrate. The 5 big ones to remember are diabetic ketoacidosis, alcoholic ketoacidosis, starvation ketoacidosis, pregnancy ketoacidosis, and salicylate overdose-induced ketoacidosis. The most commonly tested are these 3 in here, diabetic, alcoholic, and starvation. And you can tell them apart by what the blood sugar's doing. We have elevated blood glucose in a diabetic ketoacidosis, decreased glucose, hypoglycemia in alcoholic ketoacidosis, which was this case, and normal glucose in starvation. Alcoholic ketoacidosis is caused and is characterized by an underlying increased ratio of cytoplasmic NADH to NAD plus in hepatocytes, which we see because the ethanol metabolism reactions all create NADH at the expense of NAD plus. And all that increased NADH has a lot of biochemical implications, including metabolic acidoses like lactic acidosis and ketoacidosis, as well as hypoglycemia from decreased gluconeogenesis. And that's all I got for you. Good job, good luck, and keep practicing.